name is Misaki Wayengera. I'm a clinical genetist. I'm in charge of the unit of genetics and genomics within the consortium of labs that are led by Professor Moses Jolova. My biggest time is spent seeing children born with congenital abnormalities or rare Mendelian disorders. So this is one of the first labs that has been set up within the setting of Sub-Saharan Africa. It's the first in Uganda, first in East Africa. And the major future is that it's funded under the Human Heredity and Health Consortium. That's the H3 Africa Consortium, with funding particularly from the National Institute of Health and of course the Wellcome Trust as well. This is a high throughput lab that is focusing on next generation sequencing. And the key thing is for us to sequence either large sections of human DNA or the entire human genome, with the idea of understanding how changes in DNA influence disease as they interact with the environment. So the unique feature is that we have a MiSeq in here, and but the other aspect is that we are linked directly to a bioinformatics lab that you will see later. And we also have a biorepository in here. So all these features make it easy for the researchers to ship their samples. We extract DNA and then do the analysis. We run the sequencing and then the analysis is done for them as well. The key challenge that we face within the setting of the developing world for next generation, but it's not just the setting of the developing world. I think this is a global challenge, is that we cannot impact or translate what we are doing easily to the patient, because the technologies we are using are expensive. And there is a current global moratorium that is trying to aim to reduce the cost of next generation sequencing to under $1,000. But realistically, even $1,000 is quite too much for our patients. So I think that it is important that these issues are addressed, but that's a challenge that faces not just the Western world, faces us. The future that we envision is a future where we not only have MiSeq sequencing, but we move on to the advanced sequencing, the HiSeq, which is of course also becoming obsolete, and the ion torrent sequencing capabilities. And that's what we're looking at. But the other thing that we've been trying to contemplate is how possibly can we link what we are doing on research basis to patient care. We've contemplated the idea of doing the Uganda genome and sequencing the genome of Ugandans with the idea of looking at how the DNA of Ugandans impacts disease. We have interesting rare diseases like nodding disease in our setting, which are not explained fully by the environment. And we believe that it is an interaction with the environment, between the environment and DNA, that leads to these diseases, like nodding disease.